Hello everyone, my name is Mahina Akhtar and I am a biology student at Medgar College. My project is TAP1 is the new coactivator of NF-kappa B. Significance and background. How does NF-kappa B regulates myriad of genes and controls cellular response? The transcriptional activation of NF-kappa B is triggered by microbial infections, necrotic cellular agents, genotoxic and oxidative stress, and pro-inflammatory cytokines. Subsequently, the NF-kappa B directed biological functions culminate by inhibiting apoptosis, promoting cell proliferation, and stimulating angiogenesis and metastasis. Given that the NF-kappa B controls many target genes, the mechanistic knowledge of the molecular linkage between the inflammation and cancers remains incomplete. Hypothesis. How does NF-kappa B functions are regulated by transcriptional coactivators? A coactivator is a type of transcriptional co-regulator that binds to an activator to increase the rate of transcription of a gene or set of genes. The activator contains a DNA binding domain that binds either to a DNA promoter site or a specific DNA regulatory sequence called an enhancer. TAF1 is the new coactivator of NF-kappa B. We wanted to access if there are new coactivators of NF-kappa B given that it controls myriads of genes. Towards the goal, we performed a screening luciferase assay. This assay was performed by transfecting NF-kappa B promoter into the 293 T cells. As you can see, comparing 2 and 13 on the graph, it shows a big increase. Domain of transcriptional associated factor 1, TAF1. Parkinson's disease, intellectual disabilities, which are X-linked to dysmorphic features, are associated with TAF1 and also X-linked dystonia. Kinase phosphorylase the target proteins on amino acids serine, threonine, and tyrosine. Histone acetyltransferase accelerates the target proteins on amino acid lysine. Bromo domain binds to acetylated lysine on the target protein. TAF1 NTK domain activates NF-kappa B. TAF1 is a multi-domain protein, so we wanted to test which domain of TAF1 activates NF-kappa B promoter using luciferase assay. We generated the constructs for each domain. Then we transfected each domain with NF-kappa B promoter and the individual constructs. The results show that the N-terminal kinase domain of TAF1 activates NF-kappa B promoter. Kinase is a protein that functions as an enzyme to phosphorylate the target protein. TAF1 phosphorylates serine 28 on histone H3. To understand what targets are phosphorylated by TAF1, we treated the cells with TNF-alpha, LPS, IL-1 beta, IL-1 alpha, IL-6, and IL-8. The results showed that TNF-alpha phosphorylates serine 28 on the histone H3. Microarray and heat map of genes regulated by TAF1. As you can see on the right side, first THP1, second, knockdown TAF1 with RNAi. 3, treated with TNF and LPS. 4, RNA extraction. 5, microarray. 6, heat map analysis. And 7, compare with normal cells. We wanted to understand the significance of TAF1 in NF-kappa B activation, so we knocked down THP1 cells and treated the normal and TAF1 knockdown cells with TNF and LPS. Number of genes affected by knocking out TAF1 in THP1 cells. TAF wild type TNF, 54 positive, 29 negative, regulated. TAF1 knockout TNF, 36 positive, 46 negative. TAF wild type LPS, 55 positive, 29 negative. TAF1 knockout LPS, 53 positive and 30 negative. As you can see here, this shows TAF1 affects all the NF-kappa B target genes across the chromosomes, a circus plot, which shows that the NF-kappa B genes are distributed across the chromosomes from 1 to 22. TAF1 knockdown affects NF-kappa B pathways. This shows we analyzed these genes, which are a part of NF-kappa B, using the KEGG pathway. TAF1 works with transcriptional machinery to activate NF-kappa B. We proposed the model TAF1 interacts with NF-kappa B, and this shows that TAF1 regulates myriad of NF-kappa B genes. I thank the CRS program, Dean Patwari, Dr. Gautam, and my mentor, Syed Mujaba. Thank you so much.